Thank you very much. Let me just say, first of all, what a pleasure this is for me to have this opportunity to talk to you about some of our work and the issues and items and topics that I think that we share and have in common. I want to thank Dr. Wadani and Dr. Tunjung for the very kind invitation, and I very much appreciate Dean Santoso's opening remarks and comments, and the emphasis on the priorities of the food technology, and also on the importance of science education, the necessity for us to train the next generation and perhaps the generation after that, who will be the researchers who are really going to make an impact into these areas. And so I very much share those priorities, and I'm very appreciative of the opportunity to talk to you today. I will hopefully not take the whole time for my slides. I hope to leave time for questions and answers and for some discussion. But I'm also going to try not to go too quickly, because I know that maybe not everybody is an expert in bacteriophages, and there's some language and terminology which may be new or strange. Okay, now can I just check that you can see my screen okay?
Yes, it's on.
Good.
Thank you.
Okay, so the title of the talk that I gave today is From Petri Dish to Patient: Mycobacteriophages and Their Therapeutic Potential. But we are, of course, in the midst of a global pandemic where everyone has become very aware of viruses and the bad things that viruses can do. We are all struggling through these difficult times, and but are very pleased with the way that the scientific progress, I think, has brought us brought us vaccines that can hopefully allow us to move past this pandemic of the bad viruses. But I'd like to emphasize that not all viruses are bad, and there are good viruses out there. And I'm going to talk about the bacteriophages, which I would see as the viruses that can be good and can help us. I'm going to start with a reflection back to the late 2017, in pre-pandemic times, and I received a message by email from my colleague, Dr. James Soothill, who was a consultant microbiologist in London. And he told me about two patients that they have that had a very similar clinical profile. Both were young teenage women. Both had cystic fibrosis. They had both received a double lung transplant and were taking immunosuppressive drugs in order to protect the new lungs that they had received. But then both had suffered from and were suffering from bad infections with Mycobacterium abscessus, which was highly drug resistant and was not able to be controlled with many different antibiotics. And essentially, both of these patients had been sent home on palliative care because there were no other treatment options for these essentially disseminated Mycobacterium abscessus infections. And this is the text of the actual email that I received from my colleague, who said, Graham, we have two patients with M. abscessus infections which are proving hard to treat. One of the parents inquired about phage therapy. I imagine there would be considerable barriers to administering a phage, but if things get desperate, there could be a situation in which I would regard it as appropriate and ethical. So I would just sort of note that that really this was the parent of one of the patients who brought to the attention of the doctors the idea about using phage therapy, because it is not a common approach. Bacteriophages have never been used to try to treat a mycobacterium infection in a patient. And so this was very much considered as a long shot. But my colleague James had contacted me because he knew that we had worked for many years studying the bacteriophages that infect these particular types of bacteria, and therefore thought this would be a good contact to establish. And we replied and agreed that we would receive these strains and we would test them with our bacteriophages. But I never imagined that this would proceed to a therapy or anything beyond really a test of the host preferences, the bacterial preferences of the phages that we work with. So this is the one patient whose parent had made the inquiry, Isabel Carnell Holdaway, and it is mostly her story that I'll tell you about today. So as I mentioned, she had this very nasty Mycobacterium abscessus infection. It was disseminated, even though she was taking this long list of antibiotics, and also these drugs here, which are provided in order to be immunosuppressive and to support the new lungs. But what had happened was you could see much infection in the scans, and in this scan here, you can see this cross section showing a large infected node with the Mycobacterium abscessus in the liver, which is really a very bad indicator and a life-threatening condition in itself. She also had multiple skin nodules, these abscesses, all over the limbs that you can see here. They were very prominent. And the scar from the wound for the transplant was also badly infected and could not heal properly. So let me take a step back and introduce our friends, the bacteriophages, and tell you about what we've been doing to try to understand the bacteriophages, what they are, their genomics, their diversity, and then I'll show you how this proved very important for us to find phages that would be useful for a therapeutic intervention for Isabel. So bacteriophages are simply viruses that infect bacteria, often just called phages for short. Bacteriophages means bacteria eater, and so these are viruses that appear to eat, but really just infect and replicate in specific bacterial hosts. Like all viruses, they're small. They do not replicate or grow by themselves. They're absolutely dependent upon a bacterial host, and they are specific, or they have specificity for the particular bacteria that they can infect. And I will discuss more about this host preference, the bacterial preference. It can be at the level of the bacterial genus or a preference for a bacterial species, but often it is at individual bacterial strains within a species. So the host preference can be extremely narrow, and it's a very important consideration for all of the applications, whether it be in food technology or in human disease or in agriculture or in veterinary work. There are many, many different types of phages. The most abundant are the so-called double-stranded DNA tailed phages. There are others, but these are the predominant type And those are the ones that I'll tell you about today. They have a variety of behaviors or life cycles. I will not dwell too much on these, but they are important considerations. And I've shown you some pictures of these taken with the electron microscope, showing you some of their different shapes and sizes. Our colleagues that work in bacteriophage ecology have told us a lot about the population of phages out there in the environment and the world in which we live. They've shown us using these epifluorescence techniques that there is between 1 million to 10 million of individual phages or phage particles per milliliter of water or seawater. And a quick calculation tells us that the total phage population is about 10 to the power of 31 phage particles, which is an amazingly large number and tells us that phages are really the majority of all biological entities in the biosphere on the planet Earth. So they are a major part of all of our environments, and clearly we need to know more about them. The phage population is large, but it's also very dynamic as well. So there's typically maybe a 10-to-1 ratio of phages to bacteria in most environments in which we look, which means that the bacteria are being constantly infected by the bacteria, estimated at a 10 to the power of 23 phage infections per second on a global scale, which means the entire phage population turns over, in effect replaces itself on a global scale in a matter of days. It's large, it's dynamic, and it's old, with especially from structural comparisons. It's predicted that this population may go right back to the early days of life, perhaps 3 billion years or more. And so, because of these aspects of the phage population, I think it's not surprising that it's a very diverse group of viruses. There are many, many different types of genomes, many, many different types of genes, and understanding this diversity is something that's been an important consideration for us. An approach that we've taken to try to understand this question of what types of different viruses and bacteriophages are there has been to go to environmental samples, such as this picture from my garden, and to simply isolate phages, individual phages, and to purify them, and to grow them, and to characterize them, especially from a genomic perspective. And so isolating new bacteriophages is relatively simple. We take a sample, such as from this compost heap, mix it with some liquid buffer, and then plate onto a solid medium, onto Petri dishes, where we've added a lawn of a particular type of bacteria, and then we let it grow. And if a phage is present, it will infect the bacteria, it will replicate and amplify and kill the bacteria and form this area you can see here, where the bacteria are killed, and it looks like a black dot, but this is where the phage have amplified to form a plaque. This is a very old assay that was developed by Felix Durrell in the early days of phage biology, but it's nonetheless a very useful assay that we use today. Now, I told you that bacteriophages are specific for their particular hosts. And so if engaging in this process to isolate new bacteriophages, you need to choose a host that you're going to use for the isolation. And many, many years ago, we made the choice to focus on this particular strain called Mycobacterium smegmatis. The reason why we chose it was in part because it's a close relative of some of these other pathogens here. Mycobacterium abscessus, which is the strain that I introduced you at the very beginning of the talk, but also closely related to Mycobacterium tuberculosis, the causative agent of human TB. And therefore, by choosing this strain, Mycobacterium smegmatis, which is a relatively fast-growing, non-pathogenic strain, we could learn about bacteriophage diversity, but also hopefully have phages that we could put to good use for understanding and maybe even controlling these particular nasty human pathogens, Mycobacterium tuberculosis and Mycobacterium abscessus. So we were interested in isolating not just one or two phages, but many, many phages. We realized that they were very different and diverse. And so our goal was to get a whole large collection of these so that we could have a much more fuller understanding of the phage population, and essentially to try to isolate as many phages of this one particular strain of bacteria as we could. So that would be a very long and arduous project for one researcher, for one student, or one postdoctoral researcher or technician or a professor. But it can be accomplished by having many people involved, especially students who are starting out at the early stages of their careers and are interested in becoming involved in scientific research and getting engaged in the process of scientific discovery and finding new phages, new viruses, and characterizing them. And so on that premise, we developed several programs, of which the largest program is the one that is referred to as the C phages program. C has nothing to do with the oceans or the seas. It is an acronym for the Science Education Alliance, which is a group or an office, if you like, within the Howard Hughes Medical Institute, which is a major funder of scientific research and education in the United States and beyond. So C stands for the Science Education Alliance. Phages is an acronym for Phage Hunters, Advancing Genomics and Evolutionary Science. And so the idea of this program is to create a structure and an infrastructure that can support multiple institutions, universities, and colleges with many, many faculty engaged and many students. And so the program started in 2008 with 12 participating institutions, and then more colleges and universities have been added each year. There is now over 150, and these are not just research universities, the so-called R1 universities, but also other smaller community colleges and tribal colleges, because the structure provides an opportunity for everyone to participate in the phage discovery and genomics work. And so there are over 5,500 students each year, and the program continues to grow. So this is what students are doing in the program. They are starting out with their environmental sample. They're isolating the phage in the way that I showed you. They can purify and amplify to grow up large stocks, and they can purify them. They can look at them in the electron microscope. They can isolate the DNA, and then the DNA is sequenced, and then the students can participate in annotating the genomes, in identifying and predicting where all of the genes and the genetic regulatory elements are, and then looking at comparative analyses to understand the diversity, the relationships, and the evolution, and then hopefully lead to publications describing these novel findings. The program is typically delivered as 2 terms, mostly, as I'll show you, in the United States, but there is a fall or an autumn term where students are doing the phage isolation, and then a spring term where they're doing mostly the bioinformatics and the computational biology. Students are doing only maybe 4 hours per week on the project, and they usually engage as first-year undergraduate students. And so there is no pre-selection for academic excellence in some way. It is an open program where many students can discover their aptitude for doing scientific research. Now, because we have so many students and institutions all collecting these data, we have a website and a database that collects the information, but then also disseminates it as well. And it's a publicly accessible website. It's called the Actinobacteriophage database. Here is the web address, phagesdb.org. And the students get to name their own bacteriophages, and you can see some of the names that students have chosen as just illustrated as a snapshot from the homepage of the website. And then there is some information I'll show you here about the phages that have been isolated. So a total of over 18,000 individual phages have been isolated and are archived and stored in our freezers at the University of Pittsburgh. We have not sequenced all of those, but we've sequenced about 3,700, each of which has been carefully annotated and bioinformatically analyzed and then submitted into the public databases, mostly in the GenBank public database, where they can be easily accessed. And even though we have used several different bacterial species and strains for the isolation, the most relevant for this talk are those phages that infect that Mycobacterium smegmatis strain that I mentioned. We have isolated a total of about 10,000 phages, and we have complete genome sequences of over 2,000. Now, the program has some international participants in New Zealand, in Canada, in Mexico, and in Nigeria. And so the phages have been isolated. They're shown by these symbols here, have been shown on various different places around the world, but by far the majority in the United States, as I'll show you. And so this is where you are, I believe, and this is where I am. I wish that I was visiting you here, but pleased to be with you at least virtually. I'm hoping that maybe someday I will get across and actually visit you there. But the institutions are distributed all across the United States. And so you can see we have a wide distribution of places where individual bacteriophages have been isolated, and the database allows us to record all of the GPS coordinates for the individual phages that have been isolated. These have all been looked at in the electron microscope, and we see various morphotypes, but they can be grouped into these major classes that we see. You can see this one with an extraordinary long tail, this one with a contractile tail, this with a non-contractile tail, but it has this peculiar oblong or prolate capsid-shaped, in contrast to these more soccer ball shaped heads as shown here. And then there are some with little short, very short tails, and some with no tails at all. So we have many phages that look similar to each other. One of our favorites is this guy here, which has the name Mycobacteriophage corn dog, because this corn dog is a food item that's a favorite in the United States. And the student that isolated this phage was very fond of this particular food item called a corn dog. And so he thought that the phage looked just like this corn dog and therefore decided to give it its name, and is one of our favorite phages. Now, we have looked in great detail at the diversity of all of these genomes and attempted to understand that. I'm only going to give you a very brief description. The bottom line is that there are many, many different phages. We have only isolated exactly the same phage twice out of all of those sequenced genomes. But we can group the phages into those that are most similar to each other. And so there are, for example, a group of phages that we put into what we call cluster A. And so I've just circled here a set of different phages in cluster A or in cluster B, etc. And then some of these clusters can be subdivided into smaller groups, but there's many different clusters, meaning very different phages and subclusters. And here I'm just showing a phylogenetic network relationship by comparing the gene content, which genes are shared or not shared, and I'm illustrating only one example of each of the types. So you can see that these mostly radiate from a central point, indicating that these all share very, very few genes between them. They're extraordinarily diverse with lots of new genes never before discovered. And we tend to find many of those genes in some phages, but then not in others. Okay, so with this vast diversity of genomes, which have been created by this 3 billion year period of highly dynamic evolution of this large population, it's perhaps not surprising that the phages have also become finely tuned for their particular specificity for the bacteria that they infect. And I had mentioned this notion previously. It is a key item in thinking about the therapeutic utility of phages, or really any biotechnology application of the use of phages. So we would think of specificity as a double-edged sword. On the one hand, it is a very attractive facet of phages that if they only kill and infect and kill particular types of strains of bacteria, you can use them therapeutically to very finely target a particular pathogen without upsetting the rest of the microbiome in the body. Clearly, these broad spectrum antibiotics are challenging because they are often used, but they completely upset the human microbiome and can encourage antibiotic resistance of other bacteria other than the pathogen that you're trying to target. So specificity has the wonderful advantage that you can target a very specific bacterial pathogen and hunt it down and kill it with the particular phage that you want to use. However, on the other hand, specificity means that phages may only infect a very particular subset of isolates of any particular strain of bacteria. And so I'm just showing a taxonomic hierarchy here. There are certainly some phages that can infect many strains within a bacterial species, but often the specificity is right down to the strain level, or as we would think of it as the specific clinical isolates that a patient may be suffering from and that we would like to treat with bacteriophages. So specificity is a double-edged sword, but it's a key parameter and phenotype that we have to handle. So I've told you about the patient. I've introduced the patient, Isabel, who is on palliative care at this point. And I've told you that we have many, many phages and a large phage collection. The question is, do we have phages that can be used therapeutically to help this particular patient? And I'll emphasize first that the strain that we have from Isabel has our designation called GD01. So this is the very specific clinical isolate that we received from our colleague in London. And unfortunately, no one has isolated, or 3 years ago had not isolated any phages specifically for Mycobacterium abscessus. And although we tried, it turned out to be very challenging, which is why we then focused on the question as to whether there are phages in our collection which were isolated on Mycobacterium smegmatis, but which also infect Mycobacterium abscessus strain GD01 and therefore could conceivably be used for therapy. So we commonly use these so-called plaque assays, which is what I described earlier to you, where we take individual phages. I'm just showing a selection here, just a few individual phages. We can take these preparations that we store in the refrigerator. We can make serial dilutions, and in fact, these are 10-fold serial dilutions. And then we spot those onto a bacterial lawn, which you can see in this white background color. These are the bacteria. And then we incubate and we ask whether the phage can infect and form these plaques and kill the bacteria. And so this is the kind of assay that we did. This is using our control strain of Mycobacterium smegmatis, the strain on which those phages were originally isolated. And then each of those we then test on Mycobacterium abscessus GD01. And this shows one example here, a phage called Muddy. This was one that we ended up actually using therapeutically. You can see that it infects Mycobacterium smegmatis as expected, and it equally well infects GD01. I'm actually showing here two additional phages that we also used. One is this particular derivative of ZoeJ, and then this particular derivative of this phage called BPS. And I won't say too much more about them, except we had to do some additional engineering in order to get those that would be useful. So this gave us some success, although it was a rather slow process. These bacteria grow quite slowly and have to be handled carefully for their biosafety considerations. But we were pleased that after several months, we were able to identify these three potentially useful phages that we could identify as infecting and efficiently killing this particular bacterial pathogenic strain. So again, these are the three phages. One is a phage called Muddy. We could use this phage as is without having to do any further work with it. So we were very pleased with that. The second phage, BPS, that I mentioned was originally isolated in Pittsburgh many years ago. And although the parent phage itself does not work very well, we were able to engineer it and to use genetics to identify what is, in a sense, a variant which now infects and kills this strain very efficiently. So we hear a lot in the news about variants of SARS-CoV-2, such as this variant here, which appears to do much better in infecting people than the parent strain, unfortunately. But this is the same strategy and the same approach, where we can isolate variants of this phage that work optimally for infecting and killing this strain. And then ZoeJ was this third strain that we engineered so that it efficiently kills this strain. I will just add that I mentioned that there was another patient, a second patient, with a very similar clinical profile. And this strain was given the name GD02. And unfortunately, we were not able to find any phages that efficiently infect this strain GD02. And about a couple of months after we received the strains, this patient unfortunately died. These are very, very serious infections, and without a successful way of treating them, they are essentially lethal infections. And perhaps most significantly now, because of the issue of the specificity that I told you about, these three phages, which do infect and kill this particular strain, do not infect and kill the GD02 strain. And so the phages that were proved to be useful, as I'll show you for one phage, for one patient, were not helpful for the other. And that is a general issue that I'll get back to in a couple of minutes. So these are our three phages. This is what they look like. We could propagate them and amplify and purify them in the lab. We used these particular centrifugation methods to make these highly purified preparations, which you can see by these blue colored bands here. And these contain large numbers of particles. This is 10 to the power of 14 phage particles in this band that we can then pull off this particular centrifuge tube. And they're purified. We can make these preparations so they are sterile and they are free of other types of toxins and therefore suitable for administration to a patient. Clearly, we wanted to just convince ourselves that these phages actually can kill the bacteria in vitro before we tried putting them into a patient to kill the bacteria in vivo. And this is the kinds of assay that we use where we set up a grid in microtiter plates with a dilution of the bacteria alone with no phage added, or with the different amounts of bacteriophages added in these wells. And then we incubate them together for a period of time. And then we simply print that onto this agar plate and ask whether anything can grow. And so here you see the bacteria growing nicely in the absence of phage. But then this particular phage muddy has killed very nicely. These two phages also kill quite well, although you can see a little bit of survivors here at the highest concentration of the bacteria. But if you have enough phage going in, then you do get pretty good killing. And when you put all three together into a cocktail, you can now see that everything is killed. All the bacteria are killed at all concentrations that we've infected them at, exactly the profile that we would like to be able to use therapeutically. So again, this is our patient, and I told you about the clinical profile. The decision based on the advice of our clinical collaborators was that we would administer this three-phage cocktail as an intravenous administration, and we would give this twice daily at a dose of about 10 to the 9, so a billion phage particles per dose, and essentially continue to treat for as long as was needed for any clinical improvement. There was also topical administration to the skin, where there were the skin nodules, and for the sternal wound from the transplant. And as I'll show you, this was a very good outcome, in that, especially as there was no adverse reactions at all and good clinical consequences. So this issue about safety is a critical one. These preparations were being injected twice daily intravenously, and so first, do no harm, and it was very pleasing that there were no adverse reactions. This was very well tolerated, and in fact, 6 weeks after treatment, you can see that this scan showing this cross section indicates that the infected node in the liver is completely resolved. The skin nodules have largely resolved, and there is some scarring, but they're greatly improved all over the body. And then the sternal wound from the transplant is closing and healing very nicely. So the improvement is relatively slow, but very real. And Isabel has been able to return to essentially a normal life and is doing well. And this is Isabel a few months later when I got a chance to visit her in London. This is Isabel's mum, and this is Helen Spencer here, who is the physician who oversaw the treatment for Isabel. So the question that arises after this success, whether phage therapy will work with other patients with similar types of Mycobacterium abscessus infections. And so we've been sent a large number of clinical isolates, now almost 180, mostly of Mycobacterium abscessus, in order for us to test whether we have phages in our collection that infect these strains, to try to learn about this question of specificity in some detail, but then to try to help other patients if we find good phages that might be useful. I won't go into all of the details, but to summarize, we find that these bacteria, Mycobacterium abscessus, actually come in two types of colony morphotypes, the way that the bacteria appear on agar plates, so-called rough strains and so-called smooth strains. About 40% of the strains that we've received are smooth, and we have trouble finding any therapeutically useful phages for any of those strains. So that represents itself as a major blockage to proceeding with those strains. However, of the rough strains that we receive, we were able to find at least one good phage for about 80% of those strains. So not nearly as many as we would like, but it's not a hopeless number. And I'm just illustrating some of the plaque assays again that we've used as powerful tools for trying to look at the behaviors of these different types of strains. So we've had several other interventions, many of which we think have been successful and which have good outcomes. But we have one very informative case where we did not see improvement of the patient, but we think that we know why. And I think that this is an important observation. So Isabel, the patient I just showed you, was immunosuppressed with drugs to support the transplant. And we didn't see any immune response to the phages themselves. However, I'm going to show you about one patient. It's actually an older male patient with bronchiectasis and infected with this strain of Mycobacterium abscessus. We give it our strain number GD82. And this patient was treated with a 3-phage cocktail. It was the same cocktail as used for Isabel at the same dosage and the same route of administration intravenously at 10 to the 9 plaque-forming units per dose. However, this patient is immunocompetent, is not immunosuppressed at all. And what we've observed, and I'll show you, is that the infection appeared to be controlled for about a month after phage administration. And then the phage failed to continue to work. And as I'll show you, we now know that this failure of the phage infection was not because of resistance to the phages, although that could have been the explanation, but it's more consistent with the explanation that this patient mounted a robust immune response, an IgG antibody response, which not only binds to, but inactivates the phage itself. much as you would see with a really good vaccine, except we're not trying to develop a vaccine here, we're trying to deliver a therapeutic. So I'll just show you a little bit of this data. This is the patient where we're looking at the number of bacteria isolated in sputum from this patient, either before, at the time of administration of the phage, or when characterized 1, 2, 3, 4, 5, or 6 months after the start of phage treatment. And you can see here that the number of bacteria in the sputum from the lungs has dropped about 10-fold over the course of a month. However, after that, the numbers of bacteria start to go up, and they continue to increase up to the 6-month time point here. And this just shows some of the details of the immune reactions that we've looked at. This is the details of these ELISA reactions, looking at either IgG, IgM, or IgA reactions. As anticipated, the IgA specificities are very low, but then at early times, we see an IgM reaction, and then at later times, we see an IgG reaction, as illustrated here by these endpoint titers. And so the details don't matter. These are just very impressive numbers for log endpoint titers for antibodies against MUDI, against the BPS phage, and to a lesser extent, but also to the ZOE-J phage as well. So this patient at 2 months and beyond has a strong antibody response to the phages. And not only does the antibodies recognize the phages, but it also strongly neutralizes the phage activity. And so these are the kinds of plaque assays, again, that we use. And here, for example, we've taken this phage MUDI, we've incubated it with the serum from month 2, if you look right here, and then we've incubated it for a couple of hours, and then we simply ask whether the phage can still infect the strain. And here you can see the starting phage titers here, and in the 2-month and beyond sample, it has completely inactivated this phage and strongly inactivated these other 2. So this is a potent neutralizing antibody response, and the timing of it is absolutely consistent with that being the major impediment to the ongoing efficacy of the phages at 2 months and beyond. So I will finish there and just make some summary points here that, well, first of all, not all viruses are bad, and we think of phages as the good viruses, really predicated upon this notion that the enemy of our enemy is our friend. And if our enemy here are the bacterial pathogens that make us sick, then the enemy of those is going to be our friend, the good viruses that we can use for these types of purposes. In the cases that I've shown you, these are really compassionate use case studies aimed at helping individual patients. And we can certainly see a very good safety profile, and it's true in all of the patients that we've been involved in providing the phages for. But the efficacy of the phages needs to be personalized to the specific strain that a patient is infected with. And it's illustrated nicely by the differences between that GD01 and the GD02 strain. And so specificity is literally a matter of life and death in these particular cases. So strain specificity is really the key limitation in moving forward with these types of therapies. We would love to have an off-the-shelf medicine that one could simply get from the pharmacy if you have a particular type of infection. But it is this tight strain specificity which prevents us from being able to do that, at least for these Mycobacterium abscessus infections to date. Clearly, we would like to advance to a point of clinical trials where we could understand in greater detail how well the phages work, what the best or the optimal dosage is, and what the best type of route of administration is, and the overall regimen, whether the phages work together best with antibiotics or whether they antagonize each other. There is much to learn about the details of how to use phages for these kinds of applications. I'd like to thank a very large number of people who have contributed to all of this work. Especially would mention Helen Spencer, the physician I showed you earlier, who has taken care of Isabel, and our colleague Chip Schooley, who is provided a very good advice and elaboration on this particular case. The other people in my lab shown here who have advanced the work from the therapeutic perspective, and especially all of the faculty and the thousands of students, the phage hunters, who have created this amazing collection of bacteriophages, from which we were able to find a few special ones which have this ability to provide a future for a young patient such as Isabel. This community of faculty and students involved in phage research through the C-Phages program love to come together as to share our stories and our scientific discoveries. So this shows the annual symposium, which was held in 2018. Of course, we have not been able to have this over the last couple of years, but we look forward to sharing our science stories in person in the future. And I would be delighted to answer any questions that you have. So thank you. I will stop sharing and be happy to answer questions.
Yes, thank you so much, Professor Graham, for your excellent talk. We now open questions for discussion. We have around 25 minutes. If not already, I do have questions for you, actually, Professor Graham.
Good.
So I noticed that in the shapes, you have different shapes of the viruses, right? And the ones that can target the mycobacterium is the long one. So do you have any comments on that? Why do bacteriophages have different shapes? Is it related to their genome specificity, virulence, or even adaptation?
No, that's a very good question. And I'll try to not go too long on the answer, but it's a very, very good question. And so some researchers think that generally phages that have like the contractile tails, which the so-called myoviridae, are the best ones to use therapeutically. And the reason why that's often stated is because the ones with the long flexible tails, the so-called cyphoviridae, are not always, but often they are these temperate phages, which means that unless you do something about them, they don't kill the strains very efficiently. Now, and so it's not a strict correlation between the lytic infection and the shape of the phages, but there is certainly some relationship there. And in our experience, with looking at these particular bacteria, virtually all, not all, but nearly all of the phages are the ones with these long, flexible tails. So we don't really have any choice to pick the ones that people say we should have, if you understand. And so, however, it's true that most of the phages are also of this temperate nature as well. So if we just were to use them as is, off the shelf or from the refrigerator, they infect, but they don't kill the bacteria very well. And that's why we've had to do this engineering in order to try to convert them from phages that don't work very well to phages that now kill much more efficiently. And I didn't go into detail about the engineering schemes, but I think this is becoming relatively easy to do for many phages to engineer them genetically so that they have the desired properties.
I see. Oh, so I have questions already now. So
yes,
from the chat room,
yeah,
from chat. So the first one is, can we use IVIG combination to block AB? And can we use phage as combination with antibiotic to enhance therapy?
Yeah, so let me address the second question first, which is about the combination of the phages with antibiotics. And there are several key points. I think the first is that in all of the patients that we have been involved with, they have been taking antibiotics prior to phage treatment, and the antibiotic treatment continued after the addition of the phages to the treatment. And therefore, in pretty much every case, we're not looking at the treatment with only the phages, usually in combination. I think we have a lot to learn about how they work together, or if they work together. But in general, I suspect that it's the combined activity which really proves to be the most effective in the patient situation. This is a question that we would like, obviously, to address with controlled clinical trials where you can control these variables and understand the impact of the particular combinations. We are just not at that point of being able to do those trials yet, but I think that is what is needed. And then I think the first part of the question was about whether one could use sort of smart immunological interventions in order to try to block those interactions in order to prevent the neutralizing antibody response. And the question was, I think, very creatively suggesting you could develop and administer antibodies which would block the antibody response. And we've obviously not been able to try anything quite like that, but it's an intriguing and very good idea, I think. It's also possible that although it may be difficult or scary from a clinical perspective, there are some very good drugs like rituximab which inhibit that antibody response. And so although one is reluctant to add more drugs to patients that are already very sick, one could imagine strategies where you could use therapeutics to try to dampen down the immune response. I showed you the one case. In other patients, we have seen either no immune response or just a mild immune response. So again, it seems to vary from patient to patient, and we need to try to understand what causes that particular reaction.
I see. Okay, I hope that explains the question. And next one is from Fahrus. His question is, after being used as a therapy, so the phage is still in the body, and if they still exist, what are the side effects?
Yeah, so an excellent question, and again, there are many parameters that we don't have a good understanding of. Mostly, it seems that if the phages are administered intravenously, that they actually get removed from the bloodstream very quickly, within minutes or an hour, most of the phage particles are actually gone and are sort of processed out by the kidneys, etc. And so it's not, if we look in blood or in other samples, even using PCR to detect the phages, they're often very difficult to find. And if we do get a signal, it's very sort of sporadic. And so we think that what happens is the phages get cleared from the bloodstream relatively quickly, but it just takes a few particles to get to the site of the infection. And if just one or two particles of phages get to the site of the bacteria, they can then infect and amplify in the local region where the infection is. And so if you try to look in the body, you may detect phages or not detect phages, depending on whereabouts in that process the overall phage attack is happening. And so one obviously worries about when administering the phages, whether you could do any harm, whether the phages are passed off into the environment, whether they could influence the environment. Almost certainly, phages that are being released are at a very low level. And because all of these are essentially phages that occur naturally anyway, it's hard to imagine that they could be doing more harm than the ones that are present there already.
Okay, thank you. Next question is from Yusuf Wibisono. So does your group work with other actinobacteria? And in case of the Mycobacterium abscessus GD1, what was the procedure of finding the specific phages? I think he's asking a more detailed procedure than what you have explained.
So the first part of the question is whether we've worked with other actinobacteria, and so yes, as a group, but only a few other strains. So several species of Gordonia, Streptomyces, Microbacterium, Arthrobacter, and a few others that we only have a few phages for. So not many strains, but not just the Mycobacteria. And actually, we would very much like to use more different bacteria in the actinobacteria and to get more of different types of phages. In terms of the testing of the phages for those that infect Mycobacterium abscessus, I skipped over somewhat the issue that we have a very large number of phages, but we ended up finding just 3 that were useful for this particular patient. And so we did indeed, we were able to whittle it down, to narrow it down to a relatively small subset of phages that we could focus on. And in doing so, we did several things. So first of all, the genomic information, where we can sort phages into types, it provided a lot of useful information. And we knew from previous studies that phages of some genomic types are more likely to infect other strains than others. Specifically, we had taken what at the time, this was about 10 years ago, was about 200 phages, which seemed like a lot at the time. And we tested them for their ability to infect Mycobacterium tuberculosis. And we could see that some types of phages don't, and some types of phages do. So, for example, cluster B phages never infect Mycobacterium tuberculosis. And in fact, they never infect Mycobacterium abscessus. Whereas cluster K phages commonly infect Mycobacterium tuberculosis. And in fact, some, but not all, are candidates for infecting Mycobacterium abscessus. So the host range information that we had coming into this, coupled with the genomics, was able to give us some strong indicators. And so we could use that information, and then finally, because we were worried that we might be missing some information by that, we also did the kinds of experiments where we took a large collection of different phages, we pooled them to make a single mix, infected the bacteria with that mix, and then looked to see which ones were successful. And by doing that, we were able to identify at least one new group of phages that we didn't know would be able to infect, but in fact were. So I'm sorry, that's a lot of details, but that's essentially the strategy that we were able to use to pin it down to a much smaller subset than the larger collection that we have in the freezer.
I see. Oh, nice. Okay, so the next question is from Patrick. He's asking, from the plaque assay, why do you use high concentration of phages? And shouldn't the phage that cannot infect the bacteria cannot create plaque? I'm not sure what what he means by that.
Yeah, well, so I guess I would just say that the one of the nice things about working with bacteriophages is that you can work with very large numbers very easily. And so these lysates, the tubes of phage can be prepared easily at a, excuse me, at a number of 10 to the power of 10 particles per mil. So when we do the plaque assay, you can use these serial dilutions, as I showed, and you can look at infection over a very wide range. And really, when we do that, we see three phenotypes. One phenotype is where we have a phage like muddy that I showed you that infects very efficiently. There are other phages, which is the majority, where we essentially see no infection, even at the very highest titer. So when you put on, as many as probably 10 to the 8 or more phage particles, you still don't see any killing of the bacteria at all. And then there's a third group where at very high concentrations of phage, you do see some killing of the bacteria. And there are many explanations for that third phenotype. One of the reasons is that some phages give mutants at a low frequency, but can now infect the bacteria. And so that's that strategy that I described of isolating variants that now work much better. They're essentially their mutants. For other phages, we are not able to recover mutants, but we still see some killing at the very high titer. And so I'm not sure if that answers the question, but I think that sort of accounts for the strategies that we use and the phenotypes that we see.
Okay. And the next question is from Fahri. His question is related to phage therapy. So you need a lytic cycle to break the cells, right? So how do we control this particular process for them to be lytic? Is it related to quorum sensing?
So I think that the issue is mostly that I sort of skipped over this sort of central step, which is that some phages we would refer to them as lytic. They infect the bacteria, and when they infect the bacteria, there's only one outcome, which is they replicate, they kill the bacteria, and there's more phage. And so that's a predetermined, as it were, outcome. But for those other types of phages, or temperate phages, they can do the same thing, but they sometimes choose to form the lysogenic state where the bacteria survive and actually carry a copy of the phage with them. Now, that's a really important question as to which parameters influence the efficiencies of those outcomes. We don't think that quorum sensing is specifically important. I don't think we can rule it out either, so it's a really good question. But from a therapeutic point of view, the critical issue is that we know that to form lysogens, the phage has to carry a repressor gene. And so that's what we do with our engineering, is we essentially eliminate the repressor gene. So the phage starts out as being temperate, the isolate is temperate, but we then engineer it to make a derivative which now cannot form lysogens and therefore behaves just the same as a lytic phage would, where there's only a single outcome of the infection. And so we don't have to worry too much about things like quorum sensing and other signals, which for the wild type phage would play a role. A very good question, though.
Oh, interesting. Thank you. And we have another one, a question from a student also, I think. Can bacteria become resistant to phages? And if so, how do we prevent them to become a problem?
Yeah, so you have very good students with very good questions, I have to say.
Thank you.
So yeah, no, it's a very good question. So yes, resistance is a main concern, and in fact, the rationale for using the 3-phage cocktail for Isabel was because we were concerned that if we only used one phage, that we would get resistance to it, and therefore we would see a failure due to resistance. Now, for many other pathogens, E. coli, Pseudomonas, Staph aureus, etc., I think people have observed phage resistance very commonly. For these particular bacteria, to our surprise, resistance seems to happen actually at a rather low frequency, or even an undetectable frequency for many of the experiments where we've looked for it in vitro. So we could take like 10 to the 8 bacteria, we infect with phage, and then we look for survivors, and we simply don't get any at all in many instances. So resistance is a concern, although for these mycobacterial infections, it seems to be less of a concern than we had imagined. And in fact, I can't remember the exact number, but we have probably administered phages to patients where we've only had a single phage available to us that would work. We've administered just the single phage. And in each of those cases, probably 6 to 8 individual patients that have been treated this way, we have yet to recover resistance to the phages in the process of therapy. And so obviously these are more anecdotal observations, but we think that that probably resistance happens at a low frequency, but we can't rule out that when resistance, if and when resistance happens, that it may be accompanied by a loss of virulence or even increased sensitivity to the antibiotics that are being treated as well. So there's lots of room to think about trade-offs of these phenotypes where resistance could happen, but it might not be a problem clinically.
I see.
Good question.
Okay, thank you. So related to the therapy itself, the use of phages, is it approved by FDA, or is it in the pre-clinical trial that you did to the patients that you told the story to us?
Yeah. So all of the interventions and the therapies that we've provided materials for have all been done through the appropriate regulatory authorities. It's on a global scale. So some are in the United States, some are in England, in Australia, Barcelona, in Spain, all sorts of places. And so different countries have different regulatory systems. In the United States, you need approval, regulatory approval by the FDA, and there's a particular application process called an emergency IND or an EIND, where you are seeking approval on a compassionate use basis for that particular patient. And so all of the US interventions have been done with EIND approvals, with local IRB approvals as well. And then there's patient consent, and there's material transfer agreements to send the phages to the place that's going to use them. So it's pretty tightly regulated. In the UK, it's the medical research agency governs it. It's pretty strict. There's an enormous amount of paperwork and information on the use and the importing of the drugs, and so it's a pretty tightly regulated system. And we try to help provide all of the information that the regulators need in order for approval.
Okay, thank you, Professor Graham. I think our time is at the end of the session. But before we move on to our second and third talks, I would like to invite everyone to join me in thanking Professor Graham for his time and excellent talk. Please turn on your camera for a picture with him before we close the session.
That's
and
lovely.
We can get a nice picture with Professor Graham before departing.
Fantastic.
Okay.
And before we disappear, I'm sorry I can't stay on for all of the rest of the talks. It is pretty late here. But,
yes, we understand that.
But there were wonderful questions, and I would just like to thank you again for the opportunity to present the work and to discuss it with all of you students, faculty, and other researchers. So terrific.
Thank you so much. Before you leave, we'll take a picture.
Yep.
Okay.
I won't go until the picture's done.
Okay. 1, 2, 3. Okay, and then the next one. So the next. Okay, 1, 2, 3. Okay, next page. We have 6 pages.
That's good. It's good.
It's a lot. Okay. 1, 2, 3. I don't think everyone is turning on the camera, but it's fine. Okay, and page 4. 1, 2, 3. And page 5. 1, 2, 3. And the last page 6. 1, 2, 3. Okay, cool. So you can off your camera. Professor Graham, so before we leave, we would like to thank you again for your time and excellent talk. We understand that it is nighttime there, and if you have other matters to attend, please feel free to leave the seminar after this. Thank you so much for coming. And I think we can go to the next session. Professor Graham, good night, and thank you again.
Thank you.
Yeah,
thank you, Doctor.
We might invite you again in the future.
Hopefully, I can come and visit. That would be absolutely wonderful.
That would be nice. Thank you.
All right.
Okay. Bye-bye.
Bye.
Thank you. So we will now have our second talk by Dr. Agustin Krisnawardhani. Dr. Wardhani is a lecturer at Universitas Brawijaya. She received her PhD in biotechnology from Osaka University in 2007, and her research focuses on microbiology, biotechnology, and bacteriophages. And in this webinar, Dr. Wardhani will talk about the role of bacteriophages in food safety. Dr. Agustin, time is yours.
Okay, thank you, Pak Dunjung. Let me share my PPT first. Okay, can you hear my voice?
Yes, it's clear, and the share screen is also on.
Okay. Okay. Good morning, everyone. And first, I would like to thank to the team for the opportunity given to me to be able to share the topic about bacteriophages as a natural solution in improving food safety. And also thank to other speakers who took the time to share the valuable experience related to the phages and their potential. And also thank you for the committee to make this webinar happen. Okay. First, my topic will be different with the Dr. Graham talk. In this case, I would like to focus in a food safety rather than in a phage therapy. And first, I would like explain about what are bacteriophages. Bacteriophages or phages for short are the viruses that infect bacteria, and they are by far the most abundant and diverse biological entities on the planet, totally 10 the power of 31 in population. Same with the Dr. Graham said before in the previous talk. And should we worry about these bacteriophages? I think no, because these viruses are a good virus, not a bad virus, because these viruses are very hostile by recognizing the very specific receptor on the surface of bacteria. In other words, there is no harm to human, or they can undergo either lytic and lysogenic cycle. And in the food safety, we usually use the lytic cycle for the application, not the lysogenic cycle. And in the lytic cycle, the phages infected host cell, then injected the DNA into the host and finally lyse the cell. On the other hand, in lysogenic cycle, the virus will not kill the host. They will only incorporate the DNA into the chromosomal bacteria. So they cannot lyse the host. That's why we don't use the lysogenic phages to inhibit the foodborne pathogens. And let me explain in more detail between what is the difference between the lytic cycle and the lysogenic cycle in bacteriophages. Because the virus is not alive, they don't replicate through the cell division. Instead, they require host cell for the replication. And the virus, as I mentioned, have two phases for the replication. The first is the lytic cycle, come from the word lytic or lyse, which means to prick or to burst. And this is destructive or deadly cycle. And the lytic cycle of the virus will begin by the attachment of the virus on the bacteria surfaces by the proper binding protein recognition. And there is the possibility that it will get in, or at least its nucleic acid or DNA will enter into the cell. And at this point, And the primary mission of the virus is to hijack the metabolism of the cell and divert those energy resources toward reproduction of viruses rather than normal cellular function. Then the particle can get the maturity within the cell and they can be released go and infect other cells. And on the other hand, in the lysogenic cycle, after attachment to the surface cell, the genetic material of the virus will integrate into the chromosome of the host. And at this point, no replication occurs and no destruction of the host cell. It enters the nucleus and finds the nice combined spot in the DNA where it cuts the DNA and just stick it itself right in. So every time the cell divides, it's going to be carry of viral DNA from generation to generation. This is the difference between lytic cycle and lysogenic cycle. So in this case, we use the lytic cycle for the food application because it can lyse or it can burst the host cell. Means that the host cell will die after infected by the phages. And let me show you who actually discovered the bacteriophages. The bacteriophages were discovered by the British scientist Frederick Ward in 1915, and the Felix Durrell in 1917. So this is the Canadian scientist. And he was, at that time, he was trying to grow a viral strain on agar when he observed not only colonies resulting from bacterial contamination of his sample, but a strain morphology of what he assessed to be colonies of different bacterial strains. So they appear either entirely or semi-transparent, and Ford realized that these colonies were actually area of bacterial cell degeneration. So we can see in the agar plate, there is a plaque on the surface with the bacterial lawn. So the bacterial lawn means that host cell can be infected by the phages. And after that, the Felix Durrell made the independent discovery of a microorganism which appear to utilize and destroy the bacteria in the process of its own reproduction. And he called the clear areas as plaques and concluded that they were the result of a virus that attacks bacteria. So these characteristics were the basis for the Durrell naming this type of virus bacteriophages. So this is the history of the bacteriophages, a discovery of the bacteriophages. Let me move to the structure of the bacteriophages. So the bacteriophages are the bacterial virus. So the virus specific infect the bacteria. It consists the head, a collar, and the tail. The head consists the genetic material and amide of the capsomere. So the head, sometimes we call it capsid. The capsid are made of capsomere, and the function of the capsid is to protect the genome. And the next part is the next or collar. It's a short region that connect the tail to the head, and it consists many teeth. And then the third region is the tail. So this tail made up of the protein which help to injection of DNA to the host cell. So in this third region consists the base plate, to which the tail fiber and the tail fin are attached. And the function of the base plate help the attachment of the surface cell. And then the tail fibers recognize the specific cell surface receptor and attached to the cell surface. And the existence of the phages, when we isolate the phages from the sample, we can confirm by using the plaque test or the plaque assay with the double layer of agar. So in the first agar, you can use the hard agar, and after the hard agar, we can layer with the soft agar, consist the sample, and after incubation, we can see the plaque appear on the surface of agar. So it's been that we can get the phages from the sample. But if there is no plaque on the surface of agar, so we cannot get the phages from the sample. This is the assay that we usually use for isolate the phages. And how about the classification of bacteriophages? So there are many kind of bacteriophages, and however, in general, we can classify based on the morphology and the nucleic acid. Based on the morphology, we had the virus with the different head shape and also the different tail character. So some viruses or some phages have a contractile tail and non-contractile tail, and some are tailless, so they don't have tail. And also some viruses have filamentous morphology. And based on the nucleic acid, the phages can be divided into the type. So the virus or the phages which contain the double-stranded DNA or single-stranded DNA, or the phages which contain double-stranded DNA or single-stranded RNA. Oh, sorry, this is a mistake. It should be double-stranded RNA or single-stranded RNA. It's phages. So the T4 is the example for the contractile tail, and the FT is the filamentous phages, and the lambda is which have the flexible tail, and the SSP1 is the virus which have no tail. This is the classification of bacteriophages in general, but we can see many kind of type of bacteriophages. And let's move to the phages application. In the previous, I already mentioned about the lytic phages. So the lytic phages is the phages which have the capability to burst the host cell. That's why we can use the phages for many kind of application, such as the human phage therapy that already mentioned by the Dr. Graham, and also in the animal phage therapy. And also agriculture, we can use the phages to inhibit the pathogenic bacteria that make some plant, for example, tomato, pepper, have disease. So we can use the phages as the alternative solution for inhibit the kind of the pathogen. And also in other application, we can use for aquaculture phage therapy. So some fish or some fish has infected, for example, by the Vibrio. So when we use antibiotic, to handle this kind of problem, the bacteria have the resistance to antibiotic. So the phages can be the alternative solution to tackle this problem. And also for the food safety, we can use the phages for inhibit many type of pathogen. For example, Listeria, Salmonella, E. coli, etc. And also for industrial sanitation, the phages already used for biosanitizer. So instead of chemical sanitizer, some biofilm producing bacteria will contaminate the equipment in the food processing plant. So we can use phages to degrade the biofilm which attach in the equipment. So during the process, there is no contamination of the pathogen into the food. This is already done in, for example, in the USA. They apply in the dairy industry. And I will focus on the food safety for the next talk. So actually, there are many product, phage-based product already commercial. For example, Lysil. Lysil is the phage which which have the capacity to inhibit the Listeria monocytogenes. And also we have here the acryphage. This is the we can use for inhibit the bacteria, pathogen bacteria that infect, for example, tomato, pepper, and apple, and also rice. They already produce this kind of product. And also Listek same with the Lysil. This is specific for Listeria. Listeria is the pathogenic bacteria which contaminated the product, especially the refrigerated product. And the ecophage also already commercial. This is for dietary supplement. So by consume the ecophage, we have a good immunity system in our body because it will attack the bad pathogenic bacteria and maintain the good bacteria in our body. This is the example for some phage product. And how about the food application? Phages for food application. This is I would like to try to share about the condition related to the antibiotic resistance. Actually, the phages of bacteriophages is used for attack the bacteria which have the resistance to antibiotic. And this is the data show that the carbapenem resistant, the antibiotic resistant, increasing by the time until 2018. We can see there is a significant increasing of the antibiotic resistance. And also in the data show that the bacterial infection, especially the antibiotic resistant bacteria, in Indonesia, you can see that there is a huge number of bacterial infection per year. And also China, also India. And this is the problem that we face in recently. So many kind of bacteria become resistant to antibiotic. That's why the phages have the potential application for tackle this kind of problem. And we also consider about the escape pathogen. So the escape pathogen is the pathogen which have the antibiotic resistance and consists Enterobacteriophage and also Klebsiella pneumonia, Acinetobacter baumannii, and Aeruginosa, Enterobacter, and Staphylococcus aureus. So ESCAPE is the acronym for this kind of bacteria. And this kind of pathogen, it's very dangerous because they have the mechanism how to escape from the antibiotic. So they have such a mechanism that make the antibiotic cannot kill them. So the bacteriophages have the potential application to inhibit this kind of pathogen. And most of the pathogen, most of the escape pathogen, unfortunately, they can form the biofilm. So the biofilm, it's a kind of mix of the bacteria that have capability to form the matrix as a self-defense of the pathogenic bacteria. And in this case, it cannot be attacked by the antibiotic or by the antimicrobial. So it is difficult to degrade the biofilm which formed by the pathogen or the escape pathogen. That's why one alternative should be purpose that how the biofilm of the pathogen can be degraded. So the bacteriophages or phages can be a good alternative for this kind of pathogen. And this biofilm have the problem either in the surgical site, medical implant, cystic fibrosis, and also industrial masonry. So this is the biofilm problem has a big problem, I think, for this kind of food safety. And we can see here, this is the example of the biofilm. A and B is the Staphylococcus cabria and Pseudomonas fluorescens attached on the stainless steel. And the C and D, we can see the biofilm of the Bacillus cereus in the surface of stainless steel. And related to this problem, our research have conducted to cope the biofilm contamination. And the advantage used the bacteriophage as the biosanitizer or as the bio antimicrobial. It has a specific receptor and also non-toxic, and we can consider the bacteriophage safe for the human and also environmental friendly. And the bacteriophage also can enter the biofilm and lyse the host or lyse the microbe. So the bacteriophages in this case can replace the usage of the chemical sanitizer that consider to increase the microbe resistance, also have the negative impact of toxic residue and also not environmental friendly. So we try to isolate the bacteriophages from the sample, especially for the biosanitizer. In this case, we have some sample and use the Bacillus subtilis as a host. The Bacillus subtilis is the biofilm producing bacteria. So it makes some problem, especially in food industry. And from this isolation, first we confirm whether the Bacillus subtilis have the capability to form the biofilm. And from the result, we knew that the Bacillus subtilis has uh, the capability to form the biofilm. And the isolation result show that some phages already isolated. In this case, we choose the most potential phages. It means that they have the high lytic capability to the host. And the results show that PS6 is the phage we use to tackle this biofilm formation. And this figure show that the bacterial count decreased during the infection time. Means that the phages succeed or to lyse the host, in this case the Bacillus subtilis. And we can see here in the bacteriophage count, there is an increasing number of the bacteriophage during the infection time. So this means that the bacteriophages succeed in replicate and multiply the particle. So the new phages has already formed and they will burst or lyse the Bacillus subtilis. And from the isolation, we can confirm whether the phages of from the sample have the lytic cycle or the lysogenic cycle. So we can confirm by in vitro experiment. So we grow the Bacillus subtilis without phage infection, and the in the other tube we grow the Bacillus subtilis infected by the phages. So we can see the difference between I and C. I means the infected, the Bacillus subtilis infected by the virus, and C means the control. Control is the Bacillus subtilis without infection of the phages. So the infected Bacillus subtilis will result the lower turbidity compare the control. It means that the turbidity decrease because the action of the phages lysing or bursting the host cell. On the other hand, the Bacillus subtilis without infected phages will grow normal. The turbidity increase during the incubation time. We can see in the figure, in the graphic, the control and without infected phages, and the results show we infected by three kinds of phages. So the isolation, from the isolation, we've got the three kind of phages, PS6, PS8, and UA7. And another figure show that the lytic profile of the Bacillus subtilis can be different, depend on the infected time. For example, in the 30 minute, 60 minute, 90 minute, and 120 minute infected by the phages. So the different profile have shown in this figure. And the best one, the lytic profile, the best lytic profile shown by the 30-minute infected phages. So we apply this condition to infect the Bacillus subtilis to decrease the growth of the Bacillus subtilis, or in other words, we can prevent the formation of the biofilm. And other research already done. So we also isolate the bacteriophage that's specific to Salmonella. So we can see the plaque on the agar, plaque ACA and plaque ACB. So we have two kind of result, and the A result and the A plaque assay clear. It's very clear plaque. And in the B plaque assay, we can see not so clear plaque. So we can say that the B, the phages from the B plate is the lysogenic phages. However, actually we have to confirm this kind of result by a further confirmation. For example, if we want to know, or if we want to make sure lysogenic or lytic, we can see the sequence of the DNA of the phages. And the TEM image show that the bacteriophage has the head and also the tail. And for the experiment, the lysis profile of the UA2, we can see that the Salmonella specific bacteriophages have the good capability to degrade or to lyse the Salmonella. We can see that the control have the normal growth. On the other hand, when the Salmonella infected by the phages, there is no growth we can observe. We have observed the growth, but it's very low if we compare with the control. And other study also, we already get the bacteriophages that's specific to Chronobacter. Chronobacter is also pathogenic bacteria. And from this result, we can see that the bacteriophages have the high lytic activity to the Chronobacter. So we can see that the big difference, the significant difference between the control, so the Chronobacter without infection with the bacteriophage, and the Chronobacter infected by bacteriophages. And we can see also from the in vitro, from the broth, from the medium, liquid medium, we grow the control and also the control affected by the bacteriophages. So we can see the difference of turbidity. The control have the high turbidity and the chronobacter infected by bacteriophages has a clear. It means that the cell already lies or already burst by the bacteriophages. And this is the image of the key A5 of the bacteriophages, and still same with the salmonella specific bacteriophages. And this kind of bacteriophages have the head and also the tail. And from this result, we try to the host of the phages. So we try to use some host, in this case a pathogenic host. This is a salmonella, E. coli, bacillus, and listeria monocytogenes. And the results show that the phage have the lytic activity for the salmonella, bacillus, and listeria, not for the E. coli. So it means that the phage is not specific to E. coli. But from this result, we can say that the phages can be applied for food application to inhibit the salmonella, bacillus, and listeria. And as a summary from my talks today, I would like to show the advantages of phages application for food safety. So bacteriophages are highly specific and usually can infect only one species or one type of bacteria. So it means this is advantage when we use it for inhibit a certain bacteria. On the other hand, when we use antibiotic, all of bacteria will killed by the antibiotic, even the good bacteria or bad bacteria. In the case of bacteriophages, we can have a specific target to be killed by the bacteriophages. And the second one, for the advantage of the phages, there is no adverse or toxic effect on eukaryotic cell, has been observed with the use of bacteriophages. And the third, bacteriophages are ubiquitous and present in many food products and different types of soil and water sources. And phages don't change the sensory properties of food products. So there is no change related to the appearance of the food product after the food treated by the phages. And also the texture and also the color of the food cannot be changed by the usage of the phages in the food system. And the last, bacteriophages are highly resistant to the stress created during the food processing. This is some advantages of the phage application for food safety. And finally, I would like to acknowledge to some students who contribute their effort to isolate these bacteriophages. So this is undergraduate and graduate students which have worked together with me to isolate the bacteriophages. And I think this is my presentation, and thank you for your attention.
Thank you, Dr. Wardani, for your wonderful talk. And we will now have our third talk before the discussion. So the third talk will be by Dr. Hardian Susilo Adi. Dr. Adi is a lecturer at Universitas Jember. He received his PhD in molecular biotechnology from Hiroshima University in 2012, and his research focuses on plant pathophysiology and bacteriology. In this webinar, Dr. Adi will talk about the role of bacteriophages for sustainable agriculture. Dr. Hardian, time is yours. Dr.
Okay.
Agustin, if you can stop sharing, thank you.
Sorry.
Okay, thank you, Dr. Tunjung, for the invitations and introduce myself. Today, I would like to talk about slightly different, but actually the same scope of bacteriophages. In this moment, I'd like to talk about the bacteriophage in agriculture, but more specifically, I will talk about the bacteriophage in agriculture during crop production. So I'd like to share, before I start my presentations, could you hear my voice clearly?
Yes, excellent.
Okay, thank you. Maybe some part of my presentation overlap with Dr. Harful and also Dr. Agustin, but I will talk or present in another way to add some informations or share the knowledge of bacteriophages. In this case, I would like to present about the bacteriophage found in agriculture, especially in the crop productions, because we know before food and some people get a problem with the food, and then we have to start to produce the raw material of foods. So raw material of foods come from agriculture or crop productions. At this moment, I'd like to separate my talk to like four small subtopic: introduction of bacteriophage, the potential of bacteriophage in agriculture, finding, utilizing, and challenge in utilizing bacteriophage in agriculture. This is the issue in agriculture, not only in Indonesia, but also globally, because nowadays we have entered the revolution industrial 4.0, and also in agriculture, it's shifting from the conventional agriculture and to industrial agriculture. In industrial agriculture, in the crop productions, there are have characteristics. They use more widely chemical fertilizer, more chemical pesticide, that we know the impact of the use of chemical and pesticides impact on the environmental. So it is will impact on the sustainability of agriculture. So several researchers, several scholars, they suggest in sustainable agriculture, according to the combat the factor that affecting the crop productions, they can implement the IPM, especially to control plant and pest disease, plant pest and disease, like integrated pest management. So because in sustainable agriculture, people should concern on environmental, economy, and long-term practices of agriculture or crop production. I more specifically move to the integrated pest management. So in this case, more specifically talk about the plant disease caused by phytopathogenic bacteria. Normally, in this pest management, they suggest to give the priority to use the biological control agents. In this case, we can talk about the bacteriophages. Okay, this is, I think Dr. Agustin has been explained about what is the bacteria very well. So I just skip this slide, because we have known that bacteriophage is the virus that infect bacteria, or they eat the bacteria. And then, according to the International Committee on Taxonomy of Viruses, It's about 20 family of viruses, and then belongs to the bacteriophage. Okay, as well as Dr. Augustine's presentations, there are a lot of bacteriophage. They have DNA and or RNA, even single-stranded DNA or double-stranded, and it belongs to their genome. And then this is more globally morphology of bacteriophage has been found in the world. So there are about 20 families. And then in case of bacteriophage in plant disease, there are very limited bacteriophage morphology found on bacteriophage that infect plant pathogenic bacteria. I can say, according to this paper, they found only 4 or 5, but commonly only 4 bacteriophage found in agriculture, especially in crop production. For example, like bacteriophage from Myophilidae, family of Myophilidae, bacteriophage from a family of Cypophilidae, Podophilidae, and Inophilidae. This is some examples of some of my work also. We found that kind of bacteriophage in agriculture. For example, like Ralstonia solanacearum phage, this is belong to the Myophilidae, and also phage RSM3 belongs to the Inovirus, and another phage from belongs to the Cypophilidae, and also our work also in tomato plants, phage SO podovirus belong the bacteriophage that belongs to the podovirus. That all phages infect the plant pathogenic bacteria. So for the people who just who recently or just new in bacteriophage, they familiar with this picture. For example, here they know the bacteria, the virus that attach on the surface of bacterial cells and then infect it and just brush or lyse the bacterial cells. But I would like to talk more detail about this phenomenon here. So in agriculture, we found not only 2 cycles of the bacteriophage, but also there are more additional one, this chronic cycle. So this chronic cycle, as well as I found on my work, previous work during my PhD, so we found that bacteriophage, they don't kill or not follow the lytic cycle, but they follow the chronic cycle. What is the difference? What is the difference of 3 cycles of these bacteriophages? Because normally this is very common, lytic and lysogenic. In addition, found in case of Ralstonia solanacearum, we found the bacteriophages they don't lyse the bacterial cells, but they persist and produce the bacteriophage without killing bacteria. So this is called the chronic cycle. So in this case, I'd like to say and concern on the chronic cycle, because the bacteria are still alive. They can grow after some while, growth normally and not killed, because the bacteria secrete the virus, not like brush the cells and then the virus out from the cells. However, in the chronic cycle, the bacteria itself, they produce, seem like the virus actually belong to themselves. This is the chronic cycles. But in case of using the bacteriophage, as well as Dr. Augustine mentioned, that the most useful bacteriophage bacteriophage can be used as biological control in agriculture is the lytic phage. Okay, the bacteriophage with the lytic cycles. Here, I'd like to show some potential of the bacteriophage in agriculture, and has been successfully published in some papers, and continue to find the solutions how to apply this bacteriophage in our field, because we know the field has several factors that affecting on bacteriophage, and I will talk it later. Okay, what is the bacteriophage potency? So before I show the example of the bacteriophage that has been successfully tested in the bacteria that infect plants. So this is the history of the bacteriophage. Start from the thought and Darrell found the bacteriophage, and then after Alexander Fleming, he found penicillin. So the use of bacteriophage become decreased drastically, and then the use of antibiotics increased drastically. So this is the effect of the use of bacteriophage. However, these conditions after some while, and we know the impact of the use of antibiotics or the continuously or not appropriate use of antibiotics is resulting in the antibiotic resistant issue as mentioned by Dr. Augustine and Dr. Hartwell. So this one in agriculture, some of the farmers also they spray antibiotics to control the plant disease caused by the bacteria. So these antibiotics, as well as explained before, they can affect on the not only the target organism, target bacteria, but also they affect on non-target bacteria. So, and also sometimes they affect non-target living organisms, like they can accumulate in fish and the grass, and also in beneficial microorganism. Then they can kill also the beneficial microorganism in agriculture. For example, like plant growth promoting rhizobacteria, plant growth promoting fungi, and mycorrhiza, and some other microorganisms that share benefit to the plants. In 2018, one publication published that why the plant pathogenic can resist to antibiotics, because in this case they have the mechanism, the genetic mechanism they become resistant against the antibiotics. For example, Erwinia amylophora. This is the fire blight in apple. They can resist because they have a mutation on some part, and then they become resistant. And also here a lot of bacteria, phytopathogenic bacteria that has been reported become resistant to antibiotic, and they use antibiotic no longer effective to control this disease, including like the famous bacteria in rice, Xanthomonas oryza, Pathophora oryza, found in China. They found that the genetic mechanism, because this bacteria has gene AADA1 that can responsible for the resistance against the antibiotics. In another hand, the bacteria and bacteriophage also has been reported that effective on against several bacterial phytopathogenic bacteria. For example, here the bacteriophage that infect Ralstonia solanacearum, that this is the bacterial wilt disease that make the plant wilt, so they cannot produce the product anymore because they cannot grow and then die after infected by the Ralstonia solanacearum. In this case, as part of my work also, we found the bacteriophage can control this bacteria. So this bacteria cannot grow anymore, and then the tomato plant, for example, can grow and produce the tomato. And also in another case, like Xanthomonas oryza, this is also some, I also have some experience in this Xanthomonas oryza. We call this is the bacterial leaf blight on rice. This is the famous disease on the rice. And also some of the pathogen here. In the case of bacteriophage, this is the natural enemy, okay, against the plant pathogenic bacteria available in nature. We can find easily. We can find and we can collect bacteriophage from the nature, from the soil, from the irrigation water. so we can collect it because this is the natural presence as long as they target inside the in the land or in the environment. They increase number as long as the target bacterial host species are accessible, remain no residue like pesticides or antibiotics, and the more important, they sometimes more specific. They are narrow host range. So as well as I also and we our team found on in the case of Ralstonia solanacearum. This is the case, for example, one bacteriophages, they can only limit it to some group of bacteria, even they are the same species. For example, this is the case of Ralstonia solanacearum phage, RSOP1AGY. This is can infect Ralstonia solanacearum. However, they only infect the race 3 biovirtus strains. This is very specific very specific bacteriophages that infect a group of Ralstonia, but they cannot infect other groups. However, they are the same. They are the Ralstonia solanacearum. In another case, we also found Ralstonia solanacearum RSOM1 USA. This can infect not only Ralstonia species, but also they infect another sub species or subgroup of Ralstonia pseudosolanacearum. Actually, they are the same group. We call this one Ralstonia solanacearum species complex. This is species complex. They share high homology in the genome. So in this case, both phase slightly different. This is very, very specific, and this one some little bit wide spectrum, but still in the same species. Okay, next, the bacteriophage actually they most killed the bacterial host. They burst zymes because during this step, okay, in the latent period, this is the interaction period of the bacteriophage and bacteria. They still grow and produce, reproduce, and in the host bacterial cell, and then they start to lyse the bacteriophage and become start to lyse the bacterial host, and the host become brass, and then we call this one the period is brass period, and then this is the brass time. So in this case, the brass time, this depends on the dimensions of and the genome size of the bacteriophages. Normally, it's sometimes take 6 hours after one infection to lyse the cells. It takes like 6 hours and sometimes 20 minutes, depend on the size and dimension of the phage. As mentioned before, bacteriophage is very specific because they use very specific receptors. Normally, receptors of bacteriophage are flagella, pili, lipopolysaccharides. So this is why make the bacteriophage very specific, because the flagella composition, pili composition, on LPS compositions, they have different compositions, and make the bacteriophage cannot infect. Not all phage can infect all bacteria. So this is why bacteriophages become very specific. Okay. Okay, this is the example, the bacteriophage against Ralstonia solanacearum. So this is the control without the bacteriophages and without bacteriophage and without bacterial phytopathogenic bacteria. However, in wild type, this is we infect only with the Ralstonia solanacearum, the phytopathogenic bacteria. However, when we infect with another bacteriophages, so the bacterial phytopathogenic bacteria no longer can wilt the tomato plants. And when we observe also the bacteria only, this is in the case of filamentous phage, the chronic bacterial the chronic cycle phages. So they only grow, they still grow, but they cannot enter the plant. So they cannot infect and growth inside of the plant tissue. They only colonize the surface, but no longer virulent, so they cannot wilt the plant. In addition, for example, in the Xanthomonas axonopodis, the citrus canker, sometimes we found in the field. If we visit the citrus field, we found this case. This is we call citrus canker. So in this case, bacteriophage, the bacteriophage XAF and 1, they can drastically reduce the development of symptom because the bacteria, the Xanthomonas axonopodis, after infection by bacteriophage, they cannot longer grow, and then no symptoms appear on the leaf. This is in the case of our study in rice that infected by Xanthomonas axonopodis, Xanthomonas oryza, Patophora oryza, that cause the bacterial leaf blight. So also we found similar that success to control the growth of the bacterial pathogen when we use the Xanthomonas phage. In another case, against Dicea didanti, the bacteria cause soft rot on potato. We can see here that bacteriophage applications drastically reduced the number of plant death caused by the Dicea. So this is indicate the bacteriophage successfully can control the plant pathogenic bacterial and can be used in widespread in the field. So this is has been mentioned before that we can find bacteriophage easily in the environment. Sometimes I and my students, we collect the soil from the particular agriculture sites, and then we try to extract the bacteriophage from the soil, and then we isolate the bacteriophage. Then so it's not so difficult. So I think it has been mentioned by Dr. Agustin, so I will skip this slide. So in utilizing the bacteriophage, there are two ways for utilizing the bacteriophage in agriculture. We can direct applications by soil drenching, plant spraying, or indirect application by cell-infected mediated resistance. So this means we pre-inoculated first the bacterial targets, but this is slightly danger because we have to carefully, if we want to use this way in applications bacteriophage in the field. But it's not so recommended to, because we have to carefully and sure that all bacteria that we infected already infected by the bacteriophage. And the second one, utilizing the stability of particle and bacterial strain resistance are the biggest issue of phage utilization. It has been mentioned also by Dr. Hartful that some bacteria become resistant against the bacteriophage. So this is the problem during utilizations of bacteriophage. So in this case, this is the challenge in utilizing the phage for crop protection purposes. So the first one about the cells become resistant. This is the case of some work, including my teamwork also. We found that here, after bacterial cell infected with the bacteriophages, the growth of bacterial cells become decreased. But after some while, the bacterial populations become increased in growth, because we found in this case the bacterial host resistance, and they can grow even if the bacteria infected by the bacteriophage try to infect this bacteria. So because in the same tube cultures. In case of Xanthomonas also, same case, same pattern, that After infections, they can drastically reduce the growth of the bacterial cells. However, after some while, the bacterial cells become increase in number because they resistant against the bacteriophages. This is the problem when we would like to use the bacteriophage in agriculture. So it's why we have to choose or select the bacteriophage carefully while we want to use this bacteriophage for agriculture purposes. So this is why bacterial cells become resistant. There are a lot of mechanisms. This has been published also in 2018. The first one, modification of phage surface receptors. In this case, bacteria can change their receptors. They modify. So for the first time, the bacteriophage can infect. However, after infections, some bacteria they can escape and they can modify something inside the cells, and then they change they modified the cell surface, and the incoming bacteriophage can no longer infect this bacteria because they cannot find any receptors to enter the bacterial cell. In another case, we also found the resistancy mechanism because the bacteria, they have both cycles, lytic cycles in some, while they have the lysogenic cycle. This is the problem. The problem after infection, they can lyse the cell. But however, when they have the genome, in the genome, they have the gene of integrase and the phage attachment site, they can integrate the genome into the bacterial cells, and then no phage produced anymore. In this case, bacteria can still grow in the environmental. This is the problem why bacteriophages become resistant. In another case, for example, this is the restriction modifications because incoming foreign genetics, it can be restrict by restriction system in the bacterial cells. And recently, we also know that the CRISPR-Cas system found in the bacteria to protect the bacterial cells become resistant against the incoming bacteriophage. This is the new, actually not new information. This is the first time found by the researchers from the Japanese researchers, and then more detail, and two researchers, Dotna and Charpentier, they got the Nobel Prize because they concern on the CRISPR system and give impact on the genome editing. However, actually, the original CRISPR-Cas system found in bacteria to protect them from incoming bacteriophages. So, to solve the problem, by using phage cocktail, mixing bacteriophage, different bacteriophage can be one of the good choice to combat the mechanism of resistance in bacteria. And also, of course, we can use the phage with anti-CRISPR mechanism. This is the new published in the Nature. I would like to share also because this is very nice information that the bacteriophage has the mechanism to combat or to protect themselves after infection from the CRISPR-Cas system. So before move to the anti-CRISPR system in the bacteria, I would like to share some results of the research that successfully show and prove that if using cocktail bacteriophage as bacteriophage as cocktail, it can avoid from the resistancy mechanism in bacterial cell, and they can consistently reduce the growth of bacterial target. And here the example, the very new publications that they saw the bacteria has anti-CRISPR mechanism. So even if the bacteria has the CRISPR-Cas gene in the genome. However, the bacteria have the anti of this CRISPR system. So while they infect the bacterial cell, they still can assemble, produce the particle, and they protect this genome from CRISPR system, because CRISPR Cas9 system, Cas system is the enzyme that can broke the bacterial gene, broke the phage DNA inside the bacterial cells, because they have CRISPR cluster redundantly interspecies palindromic regions in their system, so they can recognize the bacteriophage, then can easily. However, when the bacteriophage in their genome has at least two protein, tubulin-like protein and cell-like protein, they can protect the genome inside the bacterial cells from incoming CRISPR system. So the bacteriophage still produce assembly and can lyse the bacterial cells. So in phytopathogenic bacteria, we also found this case. However, this result still under review in one paper under our group. So we found this mechanism found in Ralstonia also. So it's why we can use this kind of bacteriophage as good bacteriophage to control the bacterial plant disease. So the second problem is utilization, the phage stability, because we know phage are protein, are the virus covered by protein, whole their body is a protein, and then inside they have the nucleic acids. They're very unstable against the UV conditions. The problem if in agriculture, if we apply a bacteriophage in the soil, it's not so big problem. However, when we apply the bacteriophage on the phyllosphere and the upper part of plant, like, for example, like in the leaf, on the stem, so this is become a problem because the UV lights can easily destroy the bacteriophage particle. So the bacteriophage particle no longer effective against the bacterial pathogens. So this is the main problem. We can see here, for example, and also pH. So, however, normally in agricultural side, pH between 5 to 8 is still acceptable in agriculture. And also in the temperature, also more high temperature, the infectivity of bacteria decrease. In this case, the more concern is UV light, because UV can affect the bacteriophage and then reduce the infectivity, and also and even destruct the bacteriophage particles. So in this case, the suggestion is use bacteriophage in formulated form, especially for bacteriophage they are going to use on the leaf surface or plant stem surface, not in the soil. So this is the case of the utilizing bacteriophage using the micro encapsulated form. So in this case, the control, the formulated one, still stable in the environmental. However, in the free phase, it means the only phase particle in the environment, they reduce drastically because they have no protecting agent to protect them from the environment factors. Okay, this is the example of UV. In this case, UV actually to avoid the impact of the UV light. So normally bacteriophage apply in during afternoon, like 5 p.m. or 4 p.m., while the effect or the impact of the UV radiations has been drastically reduced. So this is the kind of formulations can be formed, liquid, solid, and semi-solid formulations. This is the composition normally used, commonly used in liquid formulation, for example, salt, gelatin, bovine serum, or albumin, and other materials can be used for phage formulations. Okay, this is the just example for formulation of bacteriophages. In summary, so some bacteriophages are successfully utilized as biological control in crop production. A number of research in bacteriophages that combat bacterial plant disease has been rising up recently. So it means that people and researchers start again to concern on biological or sustainable agriculture. The second, the stability of phage particle and bacterial strain resistance are the major issue in utilizing phage as biological control. So the carefully selected the phage as bacterial biological control agent is the major issue. And the second, application of phage cocktail, including the use of phage with anti-crispr mechanism, could be the best choice utilizing phage in biological control agents. And the last, in summary, formulated form help protecting phage from field applications. I'd like to thank to some institution that I have belonged to before, like study program of plant protection, Hiroshima University, SIDAS University of Jember, USDA, Department of Agriculture, and my group is research group on phage therapy in University of Jember. This is some of my students. We work specifically on bacteriophages. I think that's all of my presentations. Thank you very much for your attention.
Oh, thank you very much, Dr. Hardian, for your wonderful presentation, and Dr. Agustin. We have received a number of questions, actually quite a lot. We might not be able to cover all of them. Maybe I'll start with questions to Dr. Hardian first, and then we go to questions to Dr. Agustin. Okay. So the questions to Dr. Hardian, the first one is from Evi Kurniati. In case of indirect application, should the species bacteria cell should the species bacteria cell the same as the target? I think her question relates to the application of phages to the in the field.
Yeah, that's right. Can I try to answer? This is very nice questions, because actually this problem belongs to our recent work in my group in Bakteriophage. Okay, thank you for Evi Kurniati for the questions. In case of the indirect applications, of course we have to use the same species of or the same strain of bacterial target, because if we use another, so our bacteriophage cannot be replicate on the target. So what is the required condition? The required condition is we should, in our case, this is still in progress of our research, we make mutation of the bacterial cells. The bacterial cells become virulent, because we know the pathogen that can infect the plant only the virulent ones, the virulent bacteria. So in this case, we try to make this strain become virulent and no longer has pathogenicity. And then we just infect our bacteriophage, and then we can apply the bacteriophage, the phage infected cells into the field. So in this case, we have succeeded. Actually, this is only specific for the bacteriophage with chronic cycle mechanism. If we have another mechanism, so this is become the problem. So it's why I'm in my case, because no clear data of these applications, so I don't recommend to use this way. However, if I found through my research, recent research, so we can suggest something to apply the bacteriophage in case of indirect applications. I hope this answered your questions.
Okay, thank you. Next question is actually from me, so to you. So you mentioned that there are wide spectrum targeting ability of the bacteriophages. What actually determines that that ability, and do you see differences between bacteriophages that infect local pathogens here with the one abroad? You went to, I see your CV, you went to also the USA and also to Japan. Are there differences in the specificity of the bacteriophages that infect the probably the same bacteria?
Okay, thank you. Good questions. Actually, I have experience and I have the data that show that Ralstonia solanacearum, for example, Ralstonia solanacearum phase, they, in case of broad spectrum means the broad spectrum, they can infect more wide strain of the same species, but not another species. I mean, not another genus. For example, I have Ralstonia bacteriophage, and then they only infect strain on Ralstonia, but Ralstonia strains, not like Xanthomonas or Pseudomonas or Agrobacterium. They only specific for the Ralstonia solanacearum. However, they can infect more wide strains. And let me share a minute, a moment to show you the results here. Sorry. Okay. Okay. This is actually the. See, if we found this is RSOP1AGY is the bacteriophage found or collecting from the Egypt. So we test this strain against the bacteria, the Ralstonia solanacearum, isolated from Costa Rica, from Brazil, from Netherlands, from Kenya, Guatemala, Netherlands, Spain, and Egypt. It's give a positive result that the bacteriophage isolated from Egypt can infect all the strain collecting from another country, another continent in the world. But as I mentioned before, this is very specific only to this strain. However, also we found when we work on with Ralstonia solanacearum M1, mean the Myophyridae isolated from USA, here RSO M1 USA, they can infect from Brazil and even some country here. Guatemala, Netherlands, Taiwan from Asia, Philippines from Asia, United States from America, United Kingdom from UK, Netherlands from Europe. So it means, yeah, of course they can, because they have similarity of receptor, for example, or some other mechanism. I think it's answer your questions.
Okay, thank you. I think there's another question from the participant to Dr. Hardian. I think yeah, Lely, she's interested to know whether there are available products in the market for biopesticides, phage biopesticides.
Okay, thank you. This is nice. This is the homework for all researchers in bacteriophage in Indonesia, because until today we difficult to find it in the market in Indonesia. However, as presented by Dr. Agustin, that there is agrophage that can control bacteria, has already commercialized and available in the market, but not in Indonesia. So this is the homework of all researchers in bacteriophage to produce the product of bacteriophage and make it available in the market in Indonesia. Unfortunately, I'm so sorry.
Okay. Okay, thank you very much, Dr. Hardian. I think we can continue to Dr. Agustin now, which relates to the question before, actually, by Ali Wafa. So, what must we do to accelerate the production in Indonesia for field application, either for food or for other types of application?
Okay. I think this is our challenge. Yeah, the challenge is how we develop the phages product for the food application or agriculture application. But the first thing, we have to spread the knowledge. We have to spread the knowledge, and let's uh, work together to isolate the phages and make real. Also, Pak Hardian, please work together to make our dream come true, to make the phages a product for many kind of application. We have many sources to isolate the bacteriophages, especially Indonesia. So we have the potency for that. Why don't we go to make the product from phages for many applications? This is, I think, in Indonesia, bacteriophages not yet familiar. The research is limited. We don't have activity in huge number of the kind of research. So we can start now, from now, to make the same activity with our friend abroad, because there are many products can be found in abroad. For example, Listec, for example, Acrophage, etc. Why we can do like that? So this is our challenge actually. So let's work together.
Yes, I agree.
Yeah.
So this still about the application. There is a question from Indriani about the application in aquaculture. What do you think would be the best route? The application would it be at, oral or immersion or injection? I have no idea.
It's difficult to answer because from the point of view of the technical matters, so it is the best doing using the immersion. So treating the infected fish and using the water. So incubate, for example, 8 hour or 1 day or 2 day. I think the result is not so bad. So 90% the infected fish can be handled. But in other perspective, for example, the efficacy of the phages we can do also with the injected or oral administration also. Three of those methods we can
use all
it.
of them.
It's depend on the condition. But it is difficult to inject one by one the infected phase. I mean, the infected phase, for example, infected by Vibrio, Aeromonas, or Pseudomonas. I think that technically it's difficult.
I see.
Yeah.
And can you comment on this? Is from Fahrus. So phages that are genetically engineered, is it still safe for use in food and agriculture?
Yeah, I think why we scare with the engineered thing, because for example we eat the engineered tomato or engineered soybean. We every day we eat engineered soybean, but we still okay, right? So the same thing, if we can do the engineering of the phages, for example, you want the lytic cycle, but you don't want the lysogenic cycle, so you can engineer the gene to make them stay at the lytic cycle. I think it's okay. Engineered phages, it's not thing that we
should be worried about. Okay.
Yeah.
What do you think, Dr. Hardian, in agriculture?
Yeah.
So you mentioned that UV is a problem. So have people done engineering for UV resistant phage?
I don't see any publication of that. So as I know, only like engineered phage by modified the receptor interactions. So it can take over the problem with the resistance because the bacteria change their receptor. However, in case of UV, I'm not sure. There is the publication. So the trick is by using the formulations. It has been published actually in 2010 by a group in Florida. So they use skim milk actually to formulate the bacteriophage, and it successfully protect the bacteriophage from incoming UV light. I think that.
I will add some point, Pak Tujung, related to the lytic and lysogenic cycle. There is another way to make how the lytic cycle not become to the lysogenic cycle. I think we can manipulate the condition of the growth of the host cell. We try to make the condition to regulate, sorry, to regulate the expression of the gene of the phages. For example, the phages come to the lytic cycle because of the condition of the host cell. It's in a huge number. On the other hand, the phage will choose the lysogenic cycle when the condition of the host cell is not in a huge number. This is other way to replace the engineered modification to the phages. But I'm not sure whether is it easy or difficult to make or to manipulate the condition for the lytic and the lysogenic decision from the phages.
Okay. And another question from Shafira. Dr. Agustin, what types of bacteriophages are used commonly for food safety, and for what reasons?
As I mentioned before, that for the food application or maybe for the agriculture application, we prefer to use the lytic one, not the lysogenic one, as far as I know. Because the lytic one, it can burst, it can lyse the host cell, means that we can kill the cell, the target cell. But in lysogenic state, we cannot do like that. And the phages cannot kill the host cell. That's why it's not effective if we use the lysogenic phages. This is my opinion.
And is there any changes to the characteristics of the food from this application?
No, because this is the advantage. We use the phages. There is no change of the property of the food, even color, even texture, and even smell, etc. Because this is okay.
Yeah. So can they also resist processing conditions, Dr. Agustin? Food processing conditions.
This is also our challenge, because not all the phages can survive with the extreme condition. For example, the high pH, the low pH, on the high temperature. It's depend on the character of the phages. So this is also our challenge, how to make survive the phages that we isolate to apply in the food system. That's why encapsulation usually we use to apply the phages, for example, for biosanitizer, for biopreservative, etc. Encapsulation, and we also have to add some component that can protect the phages during the storage and during the application.
So for biosanitizers, do you have any commercial products available? This is a question from Lely.
If we talk about the Indonesia, no, we don't have at all. But if you see in abroad, for example, USA or something. I'm sure which country already have the biofilm phage for biofilm deformation.
Okay, cool.
We don't have. Yeah, we still
we
don't
still
have.
don't have.
We don't have anything.
And that leads us to the last question by Anwar Rofik. So he has a suggestion about the acceleration of production. And he has a question about BPOM. Do you think that bacteriophage application will be granted in Indonesia for application in food and or medicine? To Dr. Ardian and Dr. Agustin, any comment on that, please?
Okay. Actually, whether we will get approval from BPOM, it depends on the result of the efficiency of the phages. But in terms of the safety, I think BPOM has also, how can I say, BPOM should give the approval because the bacteriophages has no negative impact on our body. So it is nothing to worry about the bacteriophages actually, because it's specific to the bacteria, not to human, because the attachment depend on the receptor specificity. Our receptor is not compatible with the virus. So I think the bacteriophages should be approved by the people in terms of the safety and also the efficacy of the phages. This is my opinion.
Okay. What about Dr. Hardian?
Okay, thank you. I don't think so. In agriculture should pass the regulation from the BPOM because little bit different. In case of agriculture, we have like organic farming regulations. If you want to produce the biological control agents, we have follow the standard. But in my if I see in the recently we have a lot of product of biological control agents like biopesticides used by the farmers, sometimes produced by the university researchers also. They have no like quality control by the regulation, but can be used by the farmers. I think it is the same case because this is the same situations. This is biological control agents. They apply on the soil as well as the biological control agent like trichoderma and metharhizium. This is some researchers from the university. They produce and then share with the farmer, and the farmer apply on the field, and without any concern of the like organic farming regulations. But I think it will different case if the researcher try to sell to commercialize the product. This is different case because another regulations will pass by the researchers to commercialize the product. However, if only share with the farmer and the farmer accepted it to use, okay, it's not problem, I think.
Okay. Well, I think we've exhausted the time also. We have heard 3 excellent talks from Professor Graham, Dr. Agustin, and Dr. Hardian. I would like to wrap things up now by concluding with 3 main points that I noted earlier. The first one, Professor Graham, not all viruses are bad, bacteriophages are good. And his research led to the use of specific bacteriophage cocktail, native and engineered, to treat Mycobacterium abscessus infection. But he notes that a neutralizing immune response should be considered in phage therapy. And the second talk from Dr. Agustin, specific bacteriophages have been used commercially to protect food without affecting their sensory properties. For example, the use of bacteriophage to protect food from listeria, from pathogenic bacteria. And problem related to the recalcitrance of microbial biofilm can be addressed using bacteriophages as biosanitizers. However, she notes that in Indonesia, we don't have commercial products yet. Hopefully soon in the future. And the third one from Dr. Hardian's talk, bacteria have been used to control plant disease bacteria and to address antibacterial drug resistance. However, he also noted that resistance can also arise against bacteriophages. And this is partly due to receptor changes and CRISPR-Cas responses of the bacteria. And in application in the field, UV radiation also affects the effectivity of bacteriophage for applications in the field. And before we close the webinar, I would like to invite everyone to join me in thanking our speakers again for their time, excellent talk. And to close the webinar, we will have Dr. Widya Dwirukmi Putri as the head of the Department of Agricultural Product Technology to give her closing speech. Dr. Widya, time is yours.
Okay, thank you, Pak Tunjung. Good morning, everyone. I believe that during this international seminar, all of us have obtained a valuable knowledge and information from our three speakers today. We have shared their interesting and impressive work. Once again, on behalf of the Agricultural Product Technology Department, as the head of this department, I would like to give highest appreciation to Professor Headful. He already left because maybe it was already past midnight in the USA. And also to Dr. Hadian Susilo Adi from Universitas Jember or Indonesian Bakteriophage Association. Is that right? Maybe. And also Dr. Agustin Kasawardani from our department for delivering such important material so all of us can deepen our knowledge about bacteriophages. Furthermore, I would like to thank all participants who have joined this seminar. Hopefully, we can meet you again in our next seminar that will be held on next month. Thank you again, everyone. And I also thanks all the committee for handling this activity. Thank you. Wassalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam warahmatullahi wabarakatuh. Thank you, everyone, for joining us in this webinar. Thank you, Pak Ardian. I hope to meet you again.
Yeah, I hope so.
Yeah, either at Jember or here in Malang, yeah. In Jember or in Malang, ya.
Terima kasih, Pak Ardian.
Bu. Saya mohon izin live boleh nih, Pak?
Oh, iya.
Terima kasih, Pak Ardian.
Terima kasih.
Ya, terima kasih.
Terima kasih. Terima kasih.